Thanks for that lovely introduction. Can you all hear me all right? Great, great. All right. Well, today I'd like to talk to you about some of the work that I've been doing during my postdoc at Stanford. And it looks at the community assembly of the nectar microbiome. Go the other way. All right, so as ecologists, many of us are interested in why we find species where we do, how those species assemble into communities, and what those communities actually do or how they function. Now, the textbook explanation for a lot of the patterns that we see in how communities assemble in natural situations largely are deterministic. So we're taught that a species fit with its environment determines its relative its performance in that environment, and this generates a relatively predictable assembly trajectory over successional time. However, we're also increasingly recognizing that non-deterministic processes, such as those processes structured by dispersal or historical contingency, can also influence community structure and function. And these sort of rely on the ideas proposed by Gleason, by MacArthur and Wilson, and recently by Steve Hubble. Now, the microbiome is also an ecological community, so we might be able to use some of this body of ecological theory to generate some predictions about what processes structure the microbiome. I'd also like to point out that plants also have a microbiome. And many plants host very abundant communities of microbes within their roots, within their leaves, even within their stems, and in their flowers, as I'll tell you about today. And so my work examines how these microbes influence plant phenotype and plant interactions with other organisms. But one thing that I've been really interested in is why we see such extensive variation in the community composition of those microbes that associate with plants and such extensive variation in their function. And so I'd like to talk today a bit about one common theme that has emerged from our study of both animal and plant systems. And that is that we see a lot of variation among communities that occur in different sites. And so you can see here in this figure taken from a recent paper published by the Human Microbiome Project that different sites within the human body vary in their bacterial composition. So in this figure, each line represents a healthy individual, and these lines are grouped by the particular body site that they're taken from. So you may see that the sites actually differ, but what I find even more striking is that there is an extensive amount of variation within a particular site. And so I've outlined one such site here in the mouth, and you can see that among individuals, for example, firmicutes make up from about 90% of the OTUs in that site to perhaps less than 20%. So that's a huge amount of variation in the composition. And so a few commonly invoked hypotheses for this variation include potentially among person variation. They may actually just have a different mouth environment. There may also be variation in dispersal ability or arrival order within the mouth environment, or there may be historical contingency, where early arriving species actually prevent colonization by later arriving species. Now, these last two hypotheses really depend on variation in dispersal ability, variation in dispersal ability among microbial taxa. And so microbial ecologists have traditionally held that, or at least some have held, that everything is everywhere, but the environment selects. And I think we're gaining a lot of evidence that suggests that a lot of microbes do actually have a biogeography and that they can be dispersal limited. For example, in this recent study in soil fungi, for example, they've shown substantial structuring based on geographic origin, and so that's at the continent level scale. However, there have also been some recent examples studying this at a more local scale, and one particularly interesting study that looks at the effects of dispersal on the microbiome has showed that physical contact between these roller derby jammers transfers more bacteria than does air exchange between these players, and also more than the contact between the players and the rink. So needless to say, I think that more work is necessary to experimentally examine the role of dispersal in structuring the microbiome. And so that's what I'd like to talk about today. How does dispersal influence the community assembly and function of the microbiome? And so I'd like to talk today about one system in particular, and that's the community of microbes that live in floral nectar. And so we know that many plants produce this sugary exudation, known as floral nectar, that is often consumed by pollinators. However, we also find very high abundance of nectar inhabiting yeasts and also bacteria that accumulate within this resource. It's been thought that perhaps these are just commensals, or maybe they don't actually do anything, but there's been a few recent examples that show that these microbes can actually have substantial effects on both plants and pollinators. And so one example here by Rob Schaefer and Becky Irwin found that inoculation of the floral nectar with a yeast actually increased the male function, or donation of pollen-like grains onto the stigma. And so they've showed a positive effect of some of these microbes. In contrast, some work in the Mimulus system from the lab I'm working in right now has shown that if you inoculate floral nectar with a particular species of bacteria that I'll talk more about later, you actually see a decrease in pollinator foraging in those flowers, and that also reduces seed set in those plants. And so we see a lot of variation depending on the particular microbe that's found within the floral nectar. And so for that reason, it's also really important to understand what factors actually structure the composition that we see within the microbiome. So I'd like to focus in particular on one plant species today, and that's Mimulus aurantiacus. It's the sticky monkey flower. It's very common in California, occurring all along the coast in the Chaparral region. And you can see that it produces a number of abundant number of flowers. And within those flowers, we have relatively large amounts of nectar, maybe 5 microliters. It's great. And so this has been a nice system for us. So like many other flowers, Mimulus nectar begins relatively sterile, and by that I mean we haven't been able to culture any microbes from this nectar before the flower has opened. However, after the flower opens, we see an increase in the incidence of nectar samples that contain, in this case, yeast, but we also see a very similar pattern for bacteria. So there seems to be an increase in the proportion of nectar samples colonized over time. And once those either yeast or bacteria are introduced into the nectar, they grow exponentially until limited by the availability of resources. And so in our lab, or in Tad's lab, we've also shown that these nectar-inhabiting bacteria and yeast can actually compete quite strongly for those resources within the floral nectar. All right, so one likely vector for these yeasts and bacteria is the main pollinator for Mimulus aurantiacus, and that's Calypti anna, the hummingbird, Anna's hummingbird. And so just to show you that they may potentially be dispersal vectors, I'll show you a few quick results where we examined the composition of bacteria that we found on the bills of hummingbirds, and then also paired samples from nectar that was flowering at the time we caught these same hummingbirds. And so I'll just walk you through the figure very quickly. On the top, we have a bird sample, and that's actually, it represents the combined samples of about 50 different birds that we found. And so you can see that we found a lot of different taxa. So each line here represents a particular OTU that we have found. And birds actually taken in aggregate, host a lot of different bacterial taxa. And what we see is that the bacteria that we find within nectar are largely a subset of those that are found on hummingbird bills. So that's what I'd like to emphasize in this figure. And if we just compare the alpha diversity between the two, we find many more OTUs associated with birds than we do in nectar, suggesting that nectar, oh dear, that nectar itself is actually quite a harsh habitat for microbes to live in. And let me find my slide here. Okay, let's see if this works. Okay, let me see if I can talk you through the rest of it. Okay, so these pollinators are not the only visitors that come to Mimulus flowers. We also see a number of other taxa, including bees, wasps, and even thrips, potentially other sort of opportunistic nectar visitors. And these species may also spread these nectar-inhabiting microbes, particularly in light of the fact that we saw that some of these, for example, the bacteria, actually reduce pollinator visitation. And so, yeah, so we might expect that some of these other floral visitors might be might be important in mediating dispersal of some of these microbes. All right, so let me tell you a little bit more about the microbes before I go on. So both the yeast and the bacteria that we found within floral nectar seem to be very tolerant to high sugar concentrations. They can also deal quite well with the high peroxide concentrations that we find in floral nectar that plants seem to produce as a defense against these microbes. And they can also tolerate and potentially metabolize a range of secondary metabolites that are produced by the plant, as we've recently shown. But they also have significant effects on nectar itself through their growth and metabolism. They can oxidize sugars and amino acids, and also influence other properties of the nectar itself. And so I think these features and the relatively simple microbial community that we find make it a really nice system in which to study some of the effects of dispersal on the nectar microbiome. So with this main question, how does dispersal influence the community assembly and function of the microbiome? I'll focus on four questions that I'll address in sequence. First of all, how does dispersal affect abundance in the nectar? Does dispersal affect microbial species composition? Does it affect the variability that we see in the species composition and the function of the microbiome? And so I'll mainly be talking about one particular experiment that I've done out at Jasper Ridge Biological Preserve, which is in the foothills of the Santa Cruz Mountains in California. On this figure, I've noted all of the study plants that, or many of the study plants that we focus on. And I've chosen approximately 25 of those plants throughout the preserve in which to conduct the study that I'll talk about today. And those plants actually vary quite substantially in their microclimate and potentially in their genotype, although we can't separate that in this study, but we're working on that part. So you can see here among the different plants, they actually vary quite a bit in their nectar volume. Some plants produce a lot of nectar, some produce a very little, some produce, and some are quite variable in the amount of nectar that they actually produce, and some seem to always produce a lot of nectar. And this may indicate microsite variation, where some plants are in the sun and experience a lot of evaporation, for example. So on each of our focal plants, we imposed 3 different dispersal treatments. So the first was a bagging treatment intended to exclude small floral visitors and also larger floral visitors, but it presumably allowed some sort of air exchange with the environment. We also caged some flowers using a larger mesh to exclude large-bodied floral visitors. And we sampled exposed flowers that we allowed access by small and large floral visitors. And so basically we can characterize these different treatments using an increasing number of dispersal modes. So here we have basically just probably reduced wind dispersal, dispersal by smaller visitors, and dispersal by all three modes. And we did, repeated this sampling 3 times throughout the summer, and we harvested the flowers all at the same age at 5 days after they had been open. All right, so we measured pollinator visitation using one really interesting feature of Mimulus aurantiacus. So when the Mimulus flowers open, their stigma is also open. You can see that's the white part right here, the floral stigma. And when a hummingbird visits the flower and deposits pollen, or actually just touches it, the stigma closes. But it reopens if not enough pollen has been basically deposited on the stigma. And so we can actually use that as a measure of visitation or pollination, as the bees and other things actually rarely close the stigma. All right, so did our treatments work? Well, they seem to have worked pretty well. In the bagged flowers, we had very few, or basically none of the stigmas were closed. In the caged flowers, less than 20% of the stigmas were closed. And the exposed flowers had about 50% closed. So it's not a perfect treatment, but it seems to have worked to some extent. From each nectar sample, we collected microbial community abundance, basically just looking at the abundance of culturable microbes using a couple different plating or plates and media types. We also looked at the community composition using Illumina, MySeq, using the 16S, and also the ITS. I won't have time to talk about the fungal results, I'm really sorry, but I'll just talk about the bacterial community data today. We also characterize a number of the characteristics of the nectar environment, including the sugar concentration, the concentration of amino acids, and the variation in nectar volume, as that might be some sort of measure of the harshness of that particular environment. And again, we also looked at this measure of dispersal. In this case, the number of days the stigma was found closed. And so to do this, I actually visited each flower every day to look at whether the stigma was open or closed. All right, so first of all, did dispersal affect microbial abundance? And the short answer is yes. So with increasing number of dispersal modes, we found that the abundance of culturable bacteria increased. And I was concerned about perhaps pollinators only select a particular nectar type to visit. And so what I wanted to do was control for a number of other sort of environmental factors that we could control for. So I put all of these things in a model and wanted to see how well they did at actually predicting microbial abundance in nectar. And so, yes, so these particular measures here were actually summarized on a per plant basis. We used the values from the bagged flowers on those plants to sort of have a rough estimate of the initial nectar conditions which these microbes would experience, as sort of a estimate of what the environment looked like. So what we found is that most of the variation in abundance is explained by the dispersal treatment. You can see about more than 30% of the variation is explained by that. The next most important factor is the stigma age, where with increasing age of the stigma, we had a greater abundance of microbes, which is perhaps not surprising. And interestingly, we also found that the variation in nectar volume did predict some of the abundance of the microbes that we found, where flowers that actually varied to a greater extent hosted a lower density of microbes. So it seems like dispersal, followed by, to some extent, by habitat variation, explains a large amount of the microbial abundance. All right, second, we wanted to ask, does dispersal affect the species composition that we find within nectar? And so I'll present a few different types of analyses to show you what we found here. First, we'll look at an NMDS, And as other people have explained, if points are closer together, that means that those particular individual samples are more similar in their community composition. And here I'm using a Bray-Curtis metric, which takes the abundance of those particular taxa into account. And here you can see there's relatively little differentiation among the treatments. Although if you squint, you might be able to pull apart this particular group of samples. And these are, it's the only part that seems differentiated, and those are the exposed samples. However, if I recolor this figure by the abundance of microbes found in that particular sample, then I think a pattern starts to emerge. And so you can see that those particular samples are characterized by a large abundance of microbes within the floral nectar. Here I just have stigma closure, and you'll see that most of the points over here are actually triangles, indicating that that plant was actually visited by a pollinator. So it seems like that is explaining some of the variation in the community composition. If we look across these, instead of the treatments now, I have the CFU abundance, or the abundance of culturable microbes that we found in the nectar. And you can see that there seems to be a difference in the community composition that's largely driven by a couple different taxa. So if you'll see, if you can make these colors out, in the low abundance of CFU, we find quite a few, predominance of both Pseudomonas up here, and this is Burkholderia here. But with increasing CFU abundance, we have predominated, the sequence pool is predominated by Acinetobacter and Gluconobacter sequences. And just to actually do a statistical test to see which, that these particular taxa are driving this pattern, we used an analysis of differential expression, or in this case, differential OTU abundance among the 3 different treatments. And here, if the bar is above the 0 line, it means that that particular taxa was more abundant in that treatment. And so I'd just like to point out a couple different groups here. So here again is the Burkholderia, which was more abundant in the bagged and caged flowers than it was in the exposed flowers. However, you'll see that most of the other taxa that were differentially abundant were actually more abundant in the treatments where we allowed at least one or maybe multiple dispersal modes. And so here I've outlined a couple of the most obvious examples. Here again we see Acinetobacter is really seems to be very abundant in the exposed treatments compared to the others, and Gluconobacter is also very abundant in the exposed treatments compared to either bagged or caged. In addition, we found this Chrysio-bacterium, which was more abundant in the exposed treatments. And this taxa has been found to be associated with birds and may potentially be a bird pathogen, although we don't really know. The other 2 make a lot more sense. So Gluconobacter is an acetic acid bacterium, which we commonly find in nectar, and we know that it can, that it does very well in nectar. And Acinetobacter has also been described from floral nectar before. And so we expect that these particular taxa would be quite abundant. But it also seems like they rely on these pollinators for dispersal. And so did we find that dispersal affects microbial community composition? Yes, we did. And it's largely through the effects of introducing these particular taxa. All right, so then I'd like to talk about how does dispersal affect the variability in community composition. And we might predict that with increasing dispersal, or number of dispersal modes on the x-axis, we actually see a decrease in the beta diversity or variability among samples, simply because we're more likely to introduce a competitively dominant species or actually homogenize those communities to a greater extent. Is this what we find? Well, if we just looked at these dispersal manipulations, that's not actually what we find. We actually find an increase in the variability in those species pools with increasing number of dispersal modes. However, if you'll recall, we also found a lot of variation in the exposed treatment, in the exposed flowers, depending on whether or not those microbes were actually successfully introduced. So if we change this axis here to the bacterial cells per microliter, we actually see something close to the pattern that we expected, where if we have a very high bacterial density in the nectar, we see that the variability decreases. And let me give you a couple of examples to sort of illustrate this point. So here are a few examples of individual nectar samples from samples with low bacterial abundance. And you can see they're actually quite variable. The Burkholderia taxa is relatively dominant, but you can see the other species sort of pop in and out. And let me contrast this with the high bacterial abundance samples. Here we see either strong dominance by Acinetobacter or dominance by this Gluconobacter taxa. And this might be some sort of indirect evidence that we might actually see potentially some alternative community states in this case. And so does dispersal affect the variability of community composition? Well, we found that successful dispersal influences the abundance of dominant taxa, sort of homogenizing those samples, and that there might be the possibility or the suggestion of these alternative states at high bacterial abundance. All right, so what about function? Does dispersal influence the function of the microbiome? And we only looked at one particular function of these microbial communities, and we looked, in this case, at the proportion of monosaccharides, as when these microbes enter nectar, they hydrolyze sucrose to its component monosaccharides, and so we can actually see a signature of these microbes in the nectar. And so we might, and floral nectar actually starts out at about 20% monosaccharides and will increase with microbial abundance. And we see that indeed, when we increase the number of dispersal modes, we also increase this proportion monosaccharides, or the degradation of these nectar sugars. And we found that this is driven in large part by the abundance of the dominant taxa. So if you see here the proportion of sequences that are Acinetobacter or Gluconobacter, they're strongly associated with the proportion of monosaccharides in the nectar. And I shouldn't say driven, I should say associated, because we haven't, so we found strong effects when we actually look at this in the lab with Gluconobacter, but we haven't verified this with Acinetobacter. And so in answer to this final question, we found that yes, dispersal affects the function of the microbiome, and this seems to be driven in particular by the abundance of a few different taxa, mainly the specialist taxa. So in conclusion, we found that primary succession in this nectar environment seems to be limited by dispersal. We have a few other lines of evidence to support this because we have gone and inoculated flowers in the field, and we find that these yeasts and bacteria can actually grow throughout the preserve on many different plants. And we've also found a spatial signature in community similarity that I didn't have time to present. So it does seem like dispersal limitation is a key driver in this system. We also found that these specialist bacteria rely on pollinators for their dispersal. And this is kind of interesting given the fact that at least these bacteria seem to decrease pollinator foraging on samples that they've actually inhabited. And I'll finish up because I think I'm running out of time. And finally, we found that successful dispersal actually decreases the variation in species composition and generates a relatively convergent function, where we see a decrease in the beta diversity with increasing, not necessarily increasing dispersal, but increasing bacterial abundance in the nectar. Although we did see the hint that there may be a few alternative states, community states, at this high CFU abundance. So what are the implications of this work? Well, I think that it suggests that at least in these harsh harsh environments or things that are difficult to live in, that dispersal can actually be a very important driver, and particularly dispersal of the specialist microbes on which these microbial functions may actually, or microbiome functions may actually rely on. But I think that more studies are certainly needed to look at the role of dispersal across a range of different types of environments, as we may see it playing a different role in environments that are perhaps more permissive and allow the growth of a wider range of microbes. And so with that, I'd like to acknowledge a lot of people who have helped me with this work, including Tad Fukami, the PI in the lab that I'm in right now, and many other folks out at Jasper Ridge, folks in the Pay Lab, and also, my funding from the NSF, and especially the Gordon and Betty Moore Foundation. So with that, I'd like to take your comments and questions and suggestions, and thank you again for listening. If I have time for questions. Okay.
Rachel, in one of your earlier graphs, you showed that the proportion of flowers that had bacteria rose rapidly, but then it looked like it asymptoted at about 80%.
Yes.
What's going on with those other 20%?
Yeah. I think that some of those flowers are just not visited because they're separated from other plants, so perhaps the pollinators just can't find them, or they're overlooked somehow. That's my guess. Yeah, I'm not really sure. Yeah, Tim.
So I didn't totally follow, but you were saying if they are colonized, then less nectar is removed from the flower. Is that what you were saying?
So in some previous work, we've shown that if we actually inoculate the flowers with this bacteria, that pollinator foraging on those flowers is reduced. Yeah.
It's kind of a little maladaptive for them to disperse, though.
Exactly. Yeah. So this is sort of why we wanted to see if these pollinators were actually spreading some of the specialist bacteria. Yeah. Yeah, Seth.
Just a different idea. Do you think nectar is a source of antibiotic discovery because there isn't a large bacterial population then?
I think that it could be. There have been some suggestions, particularly in extrafloral nectar, that there are actually quite a few enzymes that make the environment very difficult to live in. So I think, yeah, it could potentially be. Mm-hmm. Yeah. And these bacteria and yeast are very adapted to living in a particular environment, and they exhibit a lot of very specialized adaptations for the environment. Yeah.
Have you looked at this from the plant's perspective, as in seed set or the amount of seed biomass produced?
Yes, we have. And we found that inoculation with these bacteria actually decreased seed set. We found no effect of the yeast on seed set, but it seems like the bacteria are actually negatively affecting the seed set. And that seems to be mediated through pollinator, basically pollination. But we have hints that there may be some other things going on where the flower may actually sense the presence of the bacteria and respond. But yeah, I don't have any data to show on that yet. So yeah.
How are flowers colonized each year? What's the source?
That's a great question. So we do actually see variation over time. So the communities that we find early in the flowering season are somewhat different from those that we find later in the flowering season. And it seems like the specialists really take off later in the flowering season. So we don't really know. I mean, some of these taxa have been found to be associated with insects. And so it may be there's some sort of reservoir associated with insects. I mean, they're also environmental taxa. So we find them, in the soil or on trees. So they may also be in hummingbird feeders. So we don't actually know. Yeah. A little bit, yeah. So Martin Heil has been looking at basically characterizing the defenses that are found in extrafloral nectaries. And they do find strong antifungal, basically chitinases that reduce fungal growth in extrafloral nectaries.
Are there microbes that live in that nectar
Oh,
that affect ant visitation?
very few. Even fewer microbes live in extrafloral nectar than in extrafloral nectar than in this environment. Yeah.
Okay,
All right.
well,